Hi students, this is Sayan and welcome to my biology class. Today we will discuss about restriction enzyme. From this video, we will learn about this enzyme, its mechanism and importance in biotechnology. So you should watch this video till the end. Let's get started. Restriction endonuclease, simply known as restriction enzyme, is a very important enzyme in biotechnology. At first, let's know about its definition. It is a special type of enzyme that can cleave any DNA into smaller fragments at or near specific recognition site. So this is only the simple definition of restriction enzyme. But if you want to know details about the function and importance of the enzyme, you have to know about both the term restriction and endonuclease. We have to know the exact meaning of both these terms. So let's start our discussion with the term endonuclease. What is endonuclease? Endonuclease are a group of specialized enzyme simply known as nuclease. So nuclease, a group of enzyme those can cleave nucleic acid like DNA, RNA, etc. And this group of enzyme can further be divided into two categories: exonuclease and endonuclease. Exonuclease are enzyme those can cleave any DNA from the exterior side or both of its ends. Suppose this is a stretch of DNA. Exonuclease can cut DNA from either of its end. So these are exonuclease, cut any nucleic acid fragments from any of its end. On the other hand, endonuclease are group of nuclease enzyme those can create cut near the middle of any DNA or RNA. So endonuclease are group of nuclease enzyme those are able to cut any DNA fragments near or from the middle point. Now let us know about the term restriction. What is the exact meaning of the restriction? As this endonuclease or restriction enzyme was first isolated from bacteria, so the term restriction is actually meant by their function in the bacterial cell. Whenever any foreign viral DNA enters into any bacterial DNA and try to destroy its own chromosome, then within the bacterial cell the RE or restriction enzyme destroy or stop the entry of viral DNA. Thus the actual function of this restriction endonuclease or restriction enzyme is to restrict or destroy such kind of foreign DNA into the body. But at the same time we have to know about the activity of other group of enzyme known as modification enzyme. What is this? These are group of enzyme those actually prevent the destruction of bacterial own chromosome from the activity of restriction endonuclease or restriction enzyme. So these enzyme actually bound with those sites in the bacterial DNA where restriction endonuclease may create cut. So this is the basic concept of restriction endonuclease or restriction enzyme. Now let's move to our next topic. Now let's discuss about its history or discovery. This restriction enzyme was first isolated by two scientists, Arber and Messelson, in the year of 1960. And time onward from 1960, the discovery of restriction enzyme led to the evolution of biotechnology. And from the year of 1970s, the biotechnology erupted with a huge success. So this enzyme is very important enzyme for biotechnology. Now let's know about its mechanism. Restriction enzyme cleave a DNA at a specific recognition site which consists of palindrome sequence. So for better understanding of its mechanism, at first we have to know about palindrome sequence. What is palindrome sequence? Palindrome sequence is a specific sequence on DNA whose base pair of both the strands are similar if read from a particular direction. Suppose you are reading any DNA fragments from 5' to 3' direction, and you will always have the same set of base pairs. We can think about an example. Suppose this is a segment of DNA which consists of palindrome sequence CTTAAG. This is a set of palindrome sequence. Now, if you want to read these DNA fragments from 5' to 3' direction, you will have 5' GAATTC 3'. And again, if you read this fragment of DNA of the complementary strand from 5' to 3', then you will again have GAATTC. So, such stretch of small segment in DNA are known as palindrome sequence, and palindrome sequence are a good target site for restriction enzyme. Restriction enzyme always try to digest or cut DNA fragments from palindrome sequence. This type of sequence is actually known as specific recognition sites in the definition of restriction enzymes. Now, we consider an example, and very good example of restriction endonuclease, EcoR1. This type of restriction enzyme was first isolated from Escherichia coli, the bacteria E. coli. This type of restriction enzyme actually digests this type of palindrome sequence as shown in the figure. They always digest this sort of palindrome sequence at this sequence. So, they cut the double-stranded DNA from this region, the region between G and C, and from this region, the region between G and C. So, restriction endonuclease never cut the hydrogen bond, rather they actually dissolve the phosphodiester bond at first. So, what type of cut EcoR1 may produce after its complete digestion? It will produce 5' G and AATTC 3' and the complementary strand will look like this, A and G 5'. So, here the original DNA become cut. This type of irregular shaped cut is known as sticky end or staggered cut. So, here the restriction enzyme E. coli digests the palindrome sequence and cut the double stranded DNA to produce sticky ends. So, this is how the restriction enzyme actually works. Now, let us know about the next topic. Let us discover the importance of this enzyme. Why this restriction enzyme is so important in the field of biotechnology. Actually, this type of enzyme help to produce recombinant DNA. So, let us know how the restriction enzyme help to produce recombinant DNA. Suppose it is a long stretch of DNA and we want to cut out this portion of DNA from the entire length of DNA, and for that purpose we have to take any kind of restriction enzyme. Here we are taking the restriction enzyme EcoR1, and as we all know that EcoR1 recognize this palindrome sequence to cut the DNA, it will produce a staggered cut or sticky end cut of DNA and isolate this segment of desired DNA from the entire range of the long DNA segment. So, this is our desired DNA fragments. Now, we have to introduce this DNA into any host chromosome or into any vector to clone this set of DNA. For that purpose, we have to cut the bacterial host chromosome or any cloning vector with the same kind of restriction endonuclease. So, here we are also using the restriction enzyme EcoR1, and EcoR1 is producing similar set of cut in the cloning vector or host chromosome. So, here it is the flanked region shown that is being cut by the EcoR1. Now, if we wish to produce any recombinant DNA, we have to bring this foreign DNA near this cloning vector and try to join this segment. So, let us know how this segment will get joined. This region will be placed here, A, T, T, C, and on the other hand we have to write the G, and here this segment is of foreign DNA. At the complementary strand we have to add G, then have to draw this entire stretch, and on the complementary strand we will have C, T, T, A and G. So, this segment I am depicted out from this diagram is actually of foreign DNA, and the segment of DNA drawn in the blue colored pen is actually the segment of cloning vector. Now, if we allow the joining of these two segments, we will easily have recombinant DNA. So, this is how with the help of any restriction endonuclease we can produce a recombinant DNA or genetically modified DNA. So, this is a very important mechanism in the field of biotechnology, and that is why restriction endonuclease are so important. So, from this video, hope student can get clear idea about the entire mechanism, working and importance of restriction endonuclease. So, thanks for the watching the video.